Hello, um, I am Alex, and I'm a computational biologist at Cultivarium. So we're a small nonprofit focused research organization. We're really new, and our goal is to develop genetic tools for non-model microbes and to make it easier to genetically manipulate microbes and to do open science on that general question. So there's a lot of reasons why a microbe might not be a genetically tractable. A lot of things can go wrong, and so we have projects thinking about all sorts of those things that can go wrong. To read about one of our projects on screening for replicating plasmids on bioRxiv. But today I'm going to be talking to you about a different thing that can go wrong, which is host defense rejecting your plasmid. So bacteria and archaea have natural host defense systems against foreign DNA. They don't really like foreign DNA often. There's this lovely recent meta-analysis of whole bunch of genomes and mags from the environment of different bacterial defense systems. And while there is this like recent discovery of so many cool ones, actually the one that's most abundant is still the one that's the oldest, restriction modification. And so maybe around 50 to 80% of any bacteria from a given environment can have an RM, at least one RM system, if not multiple. And what a lot of people have found in the literature is for a wide variety of microbes in the lab, restriction modification can pose a serious barrier to genetic transformation. And so there's a whole bunch of papers on all sorts of different microbes that have knocked out or otherwise circumvented restriction modification systems and found that could drastically improve, sometimes by orders of magnitude, transformation in that model in those strains. So the first thing that is on our minds with restriction modification is detecting when it's active. And so we're going to do that by looking for methylated bases with nanopore sequencing. So when the DNA goes through the nanopore, if a base is methylated, the signal changes. And so you can detect that signal change. So the question that also we were interested in is, well, do you need an unmethylated control? So there's some work out there that others have, a lot of folks have been working on this question for at least a few years now. And you'll notice that most of those papers, if you read them, compare native DNA sequence to an unmethylated control, which would be like a whole genome amplified version of the same microbe, right? And the challenge with that, though, is it does double the sequencing you have to do, right? For every strain now, you have to sequence the native DNA and the DNA without methylation, and it adds another prep as well. And because there's recently been a new pore technology for nanopore, and the accuracy has improved a lot, question in our mind was, is that really necessary now? So we have a computational workflow that we've been working on to both identify methylated sites in a genome, and then once you have those methylated sites, thinking about 5-methylcytosine and 6-methyladenosine primarily so far, then to look at those sites, gather them all, the sites that were methylated, and then identify motifs enriched in those sites, the motifs that will correspond to the methyltransferase that methylated those sites. And then once you've identified those motifs, we also then want to identify the genes in a genome that are the methyltransferase and correspondingly often the restriction enzyme. And so usually those are in an operon together. So identifying those operons. And then the final thing we might be interested in is connecting the operons you see in a genome to the motifs that you actually see methylated in that genome. So you can say, hey, this one was active, maybe that one wasn't, and to link those up. So the first thing we did to test to see if this works at all is some simple controls in a model organism, right? E. coli. So first we took an E. coli that has all of its methyltransferases knocked out, and so it should have no methylation. Indeed, in the top line there, you can see we didn't see any signal really of methylation. Then we took native E. coli K12, which has 3 methyltransferases. The first one is DAM, the second one is DCM, and the last one is DKOK1. And indeed, we could see that almost all the sites, or a majority of the sites corresponding to the known methylation specificities of those methyltransferases were appearing methylated in the native DNA. And then we had 2 more E. coli's where we knocked out or did not have ECOK1, but did have 2 heterologously expressed methyltransferases. And you can see then, we could see the methylation motifs for those 2 strains. So working well in E. coli. But we're interested more in non-model microbes. So the next place we went to was around 8 strains that had some methylation data reported for them prior. And so that was reported through the Rebase database, which is a great database put out by NEB. where they've used PacBio sequencing to call methylation in these genomes. And so I was then happy to see that the results we were getting for these strains sequenced on the nanopore was corresponding really well to the same motifs that we were seeing for each of those strains in the rebase data set. So then we could continue, and we're now going to sequence and call methylation for all our strains that did not have previously reported methylation data. And we're able to see, interesting diversity of methylation motifs, of 2 types of methylation across those strains. And it's now something that we're trying to generate data on for several more strains beyond this as well. So then the final aspect of our computational workflow then is, like I was saying, to identify the RM systems in the genome that might be linked to these motifs. And you can't always do that with confidence in cases where a genome might have, in some cases, upwards of like 10 RM systems, but you can start to make really informed guesses, which is sort of what the folks at Rebase are doing manually. For example, type 2 systems generally have motifs of certain features, as well as type 1 systems also have motifs of certain features as well. And so you can start to connect up genes that you can see in the genome to motifs based on what's known about what types of systems are likely associated with which motifs. And finally, some of the time, although not all the time, because there's only so many knowns, you can think about using homology as well, where in some cases, if a particular gene cluster has a very good hit to a known enzyme, you can make the logical assumption that, well, if at first you're unsure about 2 motifs, perhaps it's actually doing the one that it is known homolog also methylates. So where do I think things here are going in general, and what does it mean, I guess, for everyone else? I think the first thing is that definitely we'll see more work on more types of base modifications, including some of the interesting like non-canonical ones that microbes are using. It seems like you can call methylation even at low coverage. So some of our genomes, we can downsample to maybe 20x coverage, which is pretty low for an isolate, and still pretty confidently see the methylated sites and the majority of reads mapping to those sites be methylated. The other thing that you can start to do is based on if you're studying a population based on the reads that map to a given site and what % of them have methylation called for that site, you can start to estimate what fraction of the population of cells that you sequence was methylated in a case where maybe it's not all of them. I think we're moving rapidly towards a place where with nanopore sequencing as well as with PacBio sequencing, essentially you'll get this epigenetic information for free, and that can be in genomes. It can also be, as some lovely literature has shown, in metagenomes as well. And finally, the goal, at least from a synthetic biology standpoint of methylation calling, will be to then use that information to know when you should avoid RM systems in a genome, as well as to enable other approaches for avoiding or circumventing RM systems. So thank you, that's all. This is our team at Cultivarium. So you can read about and more importantly, run the code that I was talking about at our GitHub, which is github.com/cultivarium. It's not public there yet, but it probably will be in just a couple of days. And there will be like workflows that describe that, and also our results for our strains that we have too. Thank you.
Questions in the room? So how would a user interact with the company? Do we like send our isolates, or do we send the DNA, or is it like a bunch of different options?
Yeah, I would think of it the same way you would interact with a friendly academic lab, in that definitely super eager to receive strains from anyone to work with, because we're always interested in growing the collection, super eager to collaborate on projects, especially related to some of the stuff we already have going. We've been working on like a model like that with one particular collaborator already, who was very interested in getting genetic tools for a particular strain. So they just actually came on site to our offices and for a week and brought the strain with them. But also we have other friends who just send strains as well. And so definitely all of those, we're super interested in working on microbes that people actually want us to work on.
Question from online. Viruses also have restriction modification systems. Have you looked at any of them or have any plans to look into viruses?
Yeah, I'm super interested in that, especially in metagenomes, where that's where you get some of the really interesting viruses, as well as viruses that have non-canonical modifications of their DNA. So I think that's fascinating. I haven't done it myself yet, though.
Any other questions in the room?
Hi, Alex. That's really interesting. For the methylation software you're using, do you need to use a specific nanopore chemistry to get that data?
Yeah, that's a very important question, actually, because part of the motivation for working on tools here is that most of the existing tools were only working with the past 4 chemistries, and so then correspondingly, our tool will only work with our 10.4.1, which is the current chemistry moving forward. So yeah, that is a fundamental consideration.